Hello friends, welcome to my channel. Today we are going to discuss about the pollen-pistil interaction in angiospermic plant, and we will study in detail about the double fertilization. So first let's know what is the pollen-pistil interaction. So remember, the pollen grain, after its development inside the anther, after pollination, after the dehiscence, the pollen grain comes out and they travel through different pollinating agent to the surface of stigma. So remember, pollination does not guarantee transfer of right type of pollen. So every time the perfect pollen will not transfer, means compatible pollen are not always come in contact with the stigma, means the pollen grain of same species always do not land on the surface of the stigma. So remember, pistil when the pollen grain comes in contact with the pistil has the power to accept the pollen and promotes post pollination events that leads to fertilization. So remember, the pistil has the power to select or reject. When the pollen grain is of same species, it will accept, otherwise it will reject. Reject means it will prevent the pollination and germination on the stigma, so that pollen tube formation inside the style is stopped when the pollen grain is of different species or from a plant which is incompatible. Clear? So the pistil always selects the pollen grain of the same species and allow it for germination. So here we have the diagram, the LS of flower, longitudinal section of flower we have. Here we can easily see that the pollen grain germinated, the pollen grains are present at the stigma region of the pistil, because we know that the female reproductive part, the pistil, has three different parts: the stigma, where the pollen grain lands, then the style and the ovary. So the stigma receives the pollen grain, where pollen grain interacts with the pistil. So pollen-pistil interaction is seen at the surface of the stigma. When the stigma accepts the pollen grain, the pollen tube formation starts. When it rejects, no pollen tube formation takes place. So remember, when the pistil recognizes, means when it accepts, it will promote post pollination events. When it rejects, no pollen tube formation is seen. So the pollen grain germinates on stigma and produce a pollen tube. The pollen tube contains, so the content of the pollen grain move into the pollen tube. Pollen tube contains the content of pollen grain. So the pollen grain, the male pronuclei present inside the pollen grain migrate through the pollen tube. Remember, we have studied that in 60% of the angiosperms, in 60% of the angiospermic plants, the pollen grain set at two-cell stage. At two-cell stage, the pollen grain set, means the pollen grain comes out of the anther when the pollen grain has only two cells, and they are vegetative cell and generative cell. So the two cells are, the number one is the vegetative cell, and the second one is the generative cell. So the pollen grain is having vegetative and generative cell, and the vegetative cell is also called as the tube cell, because it has important role in the formation of the pollen tube. Then the generative cell divides to form two male gametes. Remember, the generative cell divides to form male gametes and another male gametes. So two male gametes are formed from the generative cell, whereas the vegetative cell do not divide, and it helps in the process of formation of pollen tube. The pollen tube enters the ovule. The pollen tube enters the ovule and it enters through the micropylar end. So the pollen tube enters into the ovule through the micropylar pole or the micropylar end, and it enters one of the synergies. So here in this structure we can easily see the mature embryo sac. This is the micropylar end where the synergids are there, two synergids are there along with the egg cell. And at the chalazal end we have three antipodals, and at the center it is the polar nuclei, two polar nuclei are present. So exactly this is situated inside the ovary. So we can see here easily all the seven cells are present here. So the pollen tube, this one is the pollen tube. It carries two cells, vegetative and generative cell. In most of the cases, two cells are there. In 40% cases, there are three cells, one vegetative and two haploid male gametes. So they travel through the pollen tube and enters into the ovule through the micropylar end. In most of the cases, they enters through the micropylar end. Remember, the filiform apparatus present here near the synergids helps the pollen tube to enter inside the synergid. So pollen tube enters with the help of the filiform apparatus to the synergids. When the pollen tube enters into the ovule, into the embryo sac with through the micropylar end, it is called porogamy. Remember, it is called porogamy. When it enters through the micropyle. So most of the cases it enters through the micropylar end. So most of the cases it is porogamy. But in some cases the pollen tube also enters through the chalazal end when it is called chalazogamy. It is called chalazogamy when it enters through the chalazal end or through the chalazal pole. Now another case is also there. Sometimes the pollen tube enters through the integument and it is called mesogamy. So it is called mesogamy when it enters through the integuments. Clear? So these are the three different cases. It depends on through which side the pollen tube enters into the embryo sac. So most of the cases it is porogamy because the pollen tube enters through the micropylar end. Okay, so all these refers to after the interaction of the pollen grain on the surface of the stigma or the pollen-pistil interaction, it will identify whether the pollen grain is of same species or not. When the pollen grain is of the same type, it will allow formation of pollen tube and migration of the two cells, the vegetative and generative, to enter into the embryo sac for further interactions. Now let's discuss about artificial hybridization. So in artificial hybridization, a breeder cross different species and even different genera, normally of different species of plants, to combine desirable character to produce commercially superior variety. So it is called artificial hybridization. So in case of artificial hybridization, it is done by the breeders to get a better variety by the artificial way to interact with the pollen grain of different species with the pistil to get a hybrid product. So in this, the superior variety is formed by artificial hybridization. It is also mentioned in your NCERT, artificial hybridization. Okay, so artificial hybridization is the method by which stigma is protected from contaminating with the unwanted pollen, and the stigma is preserved. It is not allowed to interact with the pollen grain naturally. It is protected from contamination of unwanted pollen and it is achieved by emasculation. So what is emasculation? We need to know. Emasculation is the process by which a bisexual flower, it is only done in case of bisexual flower. In this case, we remove the anthers from the bud before dehiscence. Before dehiscence of the pollen grain, we cut the anthers and remove it by forceps, and it is necessary because when we cut the anthers, it cannot self-pollinate with its own pollen grain, and we can make it by ourselves as we need. So to get a better variety, we can do emasculation, and emasculation is the removal of the anthers of a bisexual flower. It is not needed in case of unisexual flower. So emulsificated flower have covered with a bag of suitable size. The flower is covered with a bag of suitable size, which is made of butter paper. Covered with a bag which is made of butter paper. So this is covered, the flower is covered now, and it is not allowed for self-pollination as well as pollination naturally. It can only be done artificially by breeders to get a hybrid variety of plant. So to prevent contamination of its stigma with unwanted pollen grain, it is covered with a bag of suitable size which is made up of butter paper, and this process is known as bagging or bagging technique. It is called bagging technique because a bag is used in this case to cover the flower, which prevents natural pollination, and it is only done, artificial hybridization is only necessary in order to get a good or a new variety of plant. Clear? So another thing is also there, very important thing, how we can understand, how we can see the germination of pollen grain on the surface of the stigma. So this is all about the artificial hybridization. Now how we can visualize, how we can see the germination of pollen grain. So the pollen from certain types of plants like the pea plant, the pollen grain is collected and it is placed on a glass slide, and particularly one drop of sugar solution is added to the pollen grain. One drop of sugar solution, which is only 10% sugar solution, one drop added to that slide, which is having the pollen grain of plants like pea plant, and after 10 to 15 minutes, or normally it is allowed for more than 15 minutes, 15 to 30 minutes normally, because after the solution is there, after the placing of pollen grain on the solution, it is allowed for some time and washed under the microscope, under the lens, which is low power microscope, to know the detailed structure of pollen grain, and you can see pollen tube formation and germination of pollen grain. So we will take pollen grain normally from pea plant and place it on glass slide, put 10% sugar solution on the glass slide with the pollen grain and allow it for 15 to 30 minutes. Then it can be easily visualized. The pollen grain can be easily viewed, and the germination or the formation of pollen tube can be easily visualized under a microscope, which is with a low power lens. So this is how we can see germination of pollen grain under a microscope. Now we will go for the very important part, double fertilization. So now let's study about double fertilization. So double fertilization is very important for our board as well as NEET. So we will discuss about this in detail. So after the pollen-pistil interaction, after the success of pollen-pistil interaction, the pollen grain is allowed, the cells of the pollen grain allowed to enter into the embryo sac through the pollen tube. After entering the synergids, pollen tube release two male gametes. We know that the filiform apparatus help the pollen tube to enter into the synergids. After entering the synergids, the pollen tube releases two male gametes. One male gamete travels towards the egg cell. One male gamete travels towards the egg cell. egg cell and fuses with the egg cell, forming a diploid zygote. It forms a zygote which is diploid. So this is the first process and it is known as true fertilization or syngamy. This process is called syngamy or true fertilization. This is called true fertilization. Now the second male gamete travels towards the two polar nuclei. The second male gamete. So now the second male gamete, second one travels towards the polar nuclei, and it will fuse with the polar nuclei to produce a triploid primary endosperm nucleus. It will form primary endosperm nucleus, and it is having 3n number of chromosomes, so it is triploid. It is a triploid structure, and this process, due to the fusion of 3 different haploid nuclei, it is called triple fusion. 3 haploid nuclei fuses, so it is called triple fusion. It is triple fusion. So this process is triple fusion. The first male gamete travels towards the egg or the egg cell to form the zygote, which is called syngamy, which is also called true fertilization. And the second male gamete travels towards the centrally located polar nuclei and it fuses with the 2 polar nuclei, forming a triploid structure, which is the primary endosperm nucleus, and this process is known as triple fusion. Okay, so syngamy and triple fusion. So there are 2 fertilizations. The first one is true fertilization, which leads to formation of zygote. The second one is triple fusion. So as a result of 2 fertilization, the process completed. So this is called double fertilization because the 2 times fertilization takes place. So remember, the zygote after its formation develop into fetus. The zygote will develop into fetus, and after the triple fusion, the primary endosperm cell produce the endosperm which provide the nutrients. So remember, this double fertilization is a unique process in flowering plants. So double fertilization, this process of triple fusion and syngamy is only seen in case of flowering plants or angiosperms. So it is a unique feature of the flowering plants. So this is all about the interaction between the pollen grain and the stigma or the female reproductive part, the unit of female reproductive part, that is pistil. So pollen grain and pistil interact with each other. When it is found that the pollen grain is from the same species, it will allow it to travel, forming a pollen tube to enter into the embryo sac, where it releases the 2 male pronuclei, and the vegetative nuclei or vegetative cell degenerate after the formation of pollen tube, because it has important function of formation of pollen tube. That is the reason why the vegetative cell is also called the tube cell. After formation of pollen tube, it will degenerate and the 2 male pronuclei enters into the embryo sac, where the first male pronuclei travels towards the egg cell, where it fuses with the egg cell, forming the zygote with a process called syngamy or true fertilization. The second pronuclei travels towards the centrally located two polar nuclei, where they fuse together, forming a triploid structure, triploid cell known as the primary endosperm nucleus, which develop into endosperm, and this fertilization is known as triple fusion. So there are 2 fertilization. One is the true fertilization or syngamy. Second one is the triple fusion. So both together makes the process of double fertilization, which is very unique in case of flowering plant. So remember one thing, the pollen tube after formation, normally 2 cell stage occurs. After 2 cell stage, pollen grain migrate to the stigma in 60% of angiosperms. In remaining 40%, the generative cell divided into two male pronuclei, then only it will migrate to the stigma. So this is all about the process how double fertilization occurs in angiospermic plant as a result of pollen-pistil interaction. So after the interaction between pollen grain and pistil, the double fertilization completes. So this is all about double fertilization and pollen-pistil interaction. Thank you very much. If you have any doubts, please comment below. Thank you.